This is an important lesson to record because it is the review lesson, and I want everyone to have an opportunity to go back and check it out if we go over something that is unfamiliar or something with which you are uncomfortable. But I'll go back. So our objectives for the day are twofold. Biologists will be able to solidify their knowledge of the cell cycle and cell division, and biologists will be able to highlight the biggest takeaways from this unit in their notes in preparation for tomorrow's test. So I want to repeat that. There's some important points with that second objective. Biologists will be able to highlight the biggest takeaways from this unit in their notes in preparation for tomorrow's test. So if you have a highlighter, it could actually be an important time for you to use that highlighter. So that word appears there intentionally. If you don't, then there are other ways to highlight your notes with circles or underlines or stars, or just by rewriting or different colors. But I want you to be able to refer back to your notes that you've taken throughout this unit and make sure that you are highlighting the most important takeaways. And then our essential question, what should I know about cellular reproduction and how it relates to other topics in biology? So we're thinking about everything that we learned in Unit 1 about our biological macromolecules. We're thinking about everything that we learned in Unit 2 about the cell, and we're thinking about how those concepts come into play into this third unit about cell cycle and cellular reproduction. So to reiterate, today is all about review. That means no new material, but you should add to, edit, and perfect your notes based on this lesson. And what you'll see throughout the lesson is that there will be translations. So points will be made in both English and in Spanish. And what I want you all to consider is that this is an opportunity for you to ensure your notes put you in a good position to do well on tomorrow's exam. So there's nothing new. I won't be introducing any new concepts today. So you should literally be able to go through your notes page by page and edit them. So, oh, I missed that point. That's a good point. Let me add that to my notes. Or, oops, I miswrote that. That should say chromatin, not chromatid. Let me erase and rewrite that word. Or, ooh, I need to make sure that I emphasize this point. Let me highlight it. Okay, so that's what today is about. And I'm going to ask that you all contribute to the conversation and are engaged as much as possible as well. I have some questions to ask you. All right. So we've got two standards in Unit 3 that are tested on the end of course exam. We've got standard 1.2.2, and we also have standard 3.2.1, which we'll talk about later in today's lesson. But standard 1.2.2 is all about the cell cycle. And it states that students should be able to analyze how cells grow and reproduce in terms of interphase, mitosis, and cytokinesis. So what you'll also notice today is that I don't highlight anything. I'm not highlighting anything. So I'm not telling you exactly what you need to write down. It's up to you to interpret what you need to add to your notes. Again, there's nothing new. This is also a good practice for those of you who are college bound, because when you're in a college lecture, the professor will not tell you exactly what you need to write down. In fact, if you go and you don't have anything in front of you at all during the entire lecture, no one's going to say anything to you. And if you go and you try your best to speed write literally everything that you see, again, no one will say anything to you, but you're going to end up with more information than you need, and it could end up confusing you. So best practice as we think about advancing in our academic careers, note-taking should be efficient and effective. Efficient meaning it doesn't take too much time or energy. Effective meaning that you cover all of the important concepts. All right, so I'm not highlighting anything for you all to write down today. That's up to you to decide. You can always pause me and ask me a question or type a message to the chat if you need me to go over something though. All right. So it may be a good idea to write this in your notes if you haven't already. The cell cycle, what's the first part of the cell cycle? Assuming we start with interphase, what will be the first part?
Interphase.
Yes, and what's the first part of interphase, Kiki? Or anyone else? And thank you, Kiki, for showing yourself. Does anyone know what the first part of interphase is? Come on, guys. This should be a quick.
Growth 1.
Growth 1. Thank you. Also known as G1 or gap 1. But Alicia, you are 100% correct. And during gap 1, we know that the cell grows. Okay, so just moving quickly because we have a lot to cover today. What comes after growth 1 or gap 1 or G1, whatever you want to call it?
Synthesis.
Excellent. Synthesis. And what happens during synthesis?
Cell replication.
Excellent. More specifically, DNA replication. The DNA is replicated during synthesis. We're synthesizing new DNA. We're making new DNA. What about after synthesis?
Gap 2.
Excellent. Gap 2. Again, that's a period of more cell growth. I know this feels like we're moving quickly, but again, no new information here. This was covered a week ago today on day 1 of our unit. So if you need to write anything down, I know you're probably moving quickly, but it should also be somewhere in your notes previously. What comes after gap 2?
Cytokinesis.
And there's something right before that. There's something before cytokinesis. But thank you, Gabby.
Meiosis or mitosis.
Yes, the M phase, which could be mitosis or meiosis. And then, as Gabby said, we have cytokinesis. And after cytokinesis, we end up with 2 new cells or 4 new cells. And those cells will re-enter the cell cycle themselves, which is why it's called a cycle. Keep going. All right. So my question for you then, why are G1, S, and G2 highlighted in yellow? So if you can, let me get my pointer. You see these 3 steps are highlighted in yellow. Why is that?
Is it because it's the most important phase in the cell cycle?
Okay, so it is a very important phase, especially synthesis, which is when the DNA is replicated. Any other reason? Why would they be highlighted? It's the longest. And so what is that longest phase called?
Interphase.
Interphase, excellent. So they're highlighted yellow because they all represent the same, they're all parts of the same phase. They're all parts of interphase. Good job. So I'll leave this up for 15 seconds and you all can pull out as much information as you need. Okay. So true or false. So I want everybody participating in this part of the class. So we're going to go through, I think there are 4 questions. I want you to type into the chat, true or false. Okay, depending on, whatever you think the correct answer to the question is. So true or false, mitosis is the longest phase of the cell cycle.
False.
Good. I see a lot of falses. Excellent. I see a flosse. Good. All right. That is correct. That is a false statement. And interphase is the longest phase of the cell cycle. So we would need to replace that word mitosis with interphase. Good job. Okay, true or false? The DNA must be replicated before the cell divides. Is that true or false? Good. I see a lot of trues coming in. Good, good, good. Excellent. Yes, that is a true statement. The DNA is replicated before M phase, and the DNA gets replicated during synthesis. Okay, so seems like you guys have a good grasp on that question. Okay, true or false? Cytokinesis is the process of two cells fusing together. True or false? Cytokinesis is the process of two cells fusing together. Okay, I see a lot of falses, and I see an IDK. So can someone help Angel out? Why do they think that is a false statement? How could it be corrected to be true?
They don't fuse, they divide or duplicate in the end.
Excellent. Thank you. So cytokinesis does not mean fusing together. It actually means splitting. Okay. So cytokinesis is the process of one cell splitting into two different cells. So that was a false statement. So good job on that. One more. Oh, no, that was the last one. Sorry. All right. So you guys have a good grasp on the cell cycle in general. And we know that the second phase of the cell cycle, mitosis, can also be broken down into four of its own phases: prophase, metaphase, anaphase, and telophase. And one of the essential skills within this standard 1.2.2, which is about the cell cycle, is that students should be able to identify different steps or different phases of mitosis based on pictures. All right, so you all should start thinking, and we're going to go over it over the next 4 slides, about how you can identify which step of mitosis we're talking about just by looking at pictures. That will be something that you are asked to do on tomorrow's test. So this first stage of mitosis is called prophase. During prophase, the nuclear membrane dissolves, the DNA condenses into chromosomes. So instead of being that big plate of spaghetti where all the DNA is just totally and randomly wound together, the DNA condenses, it forms into tightly wound chromosomes that are extremely well organized. And the spindle fibers, which here look like these yellow cords, the spindle fibers move into place. We can see that they get attached to the chromosome. So how can you identify this picture as prophase? What tells you, what in this picture tells you that it's prophase? What is a clue? That
It
this
still
is,
looks intact.
say that one more time.
It still looks intact.
Okay, yeah, we can still see that the chromosomes are still intact. Is that what you meant?
Yeah.
Okay, good. That's true. That's a good point. What else? It looks like there is a white, and maybe it's hard for you all to see on your screens, but there's a white dotted line here, or white dotted lines. What is that representing?
The nuclear membrane.
Excellent. So this first point here says that the nuclear membrane dissolves. So it's literally starting to just break apart. And the nuclear membrane is like the outer covering of the nucleus. So in this case, we can see that these dotted lines kind of represent how it's just breaking apart. The nuclear membrane is breaking apart. And that gives the spindle fibers access to the chromosomes. If the nuclear membrane was still intact, these spindle fibers would not have access to these chromosomes. They would be blocked by the nuclear membrane. But now that the nuclear membrane is dissolving, the spindle fibers can start to attach to the chromosomes. The next stage is metaphase. And this one is much easier to identify because the chromosomes line up in the middle of the cell. And you can also see that the spindle fibers are attached to the centromeres of the chromosomes. So there's a lot. That was a lot of jargon right there. The spindle fibers are attached to the centromeres of the chromosomes. We can see that these spindle fibers, the yellow cords, are literally being attached to the middle of these chromosomes, and that's called the centromere. So metaphase, how can we identify that this picture is metaphase? Again, this one should be pretty easy.
Can you repeat that again?
Yes. How can we identify this picture as metaphase?
The
The
chromosomes are lined
chromosomes are in
up.
the middle.
Exactly. Yeah. Metaphase is probably the easiest to identify because you literally have the chromosomes lined up in the middle. The next stage of mitosis is called anaphase. Anaphase. This is when the sister chromatids are pulled apart and they move towards the poles, aka the ends of the cell. So here they were in the middle. Now they're being pulled towards the ends of the cell. Middle, pulled towards the end. Middle, pulled towards the end. So how can we identify this picture as anaphase? What tells us, what's a clue that this is anaphase?
When the chromosomes are split.
Yes,
Pulled apart.
yes, exactly. We can see that the chromosomes have literally been pulled apart, right? So instead of them being an X, we get one half being pulled this way and the other half being pulled this way. We also talked on Tuesday of last week about ana, the prefix ana meaning apart. So anaphase is the step when our chromosomes get pulled apart. That should be your clue there. And lastly, telophase. This is when the nuclear membrane begins to reappear. The DNA again collapses into chromatin. So instead of being in these nice chromosomes, which look like X's, now it collapses again into these random unorganized strings of DNA called chromatin. And the cell also begins to cleave into two new cells. So again, last one, how can we identify this picture as telophase? What's the clue? What are the clues?
You can see the nuclear membrane to like start showing up again.
Excellent, Ayana, yes. You can see that these white dotted lines are back, and that represents that the nuclear membrane is starting to reform. Anything else?
The chromatids are split.
Good, yeah. We've got chromatin that's reappearing. We don't have any chromosomes in this picture. And then probably the biggest hint or clue that this is telophase is we can literally start to see there are two new cells being formed. Here's a cell and here's a cell. And we know that right after telophase, we end up with cytokinesis. So these two cells will fully split. And you end up with two distinct cells with their own with their own plasma membranes. All right. So, it's important for you to be able to identify each of these phases or each of these stages of mitosis based on pictures. And you will, I can guarantee you will be asked to do that on tomorrow's test. Any questions about that before I move on? Thank you, Angel. I saw Kiki shake her head. All right. Okay, so characteristics of mitosis. And stop me at any point if you have questions about this process. Mitosis only has one round of prophase, metaphase, anaphase, and telophase. It only goes through each of those things twice. Sorry, it only goes through each of those things once. Sorry about that. And we can see here, I can literally see, here's the cell. This is interphase, so the cell has just replicated its DNA. It's gone through 2 gaps or 2 growth stages. Here's prophase, so the nuclear membrane, which the nucleus here is in yellow. I don't see any yellow in this picture. So the nuclear membrane has now dissolved. Here's metaphase. I can tell because the chromosomes are lined up in the middle. Here's anaphase as they begin to get pulled apart. I could say that both of these stages are well, let's call this telophase, and this is cytokinesis. So it only goes through each stage, prophase, metaphase, anaphase, telophase, once. Now the cell gets pulled apart and we eventually end up with 2 daughter cells. So that's the second point. Mitosis produces 2 daughter cells, and we can see it here. One cell, after prophase, metaphase, anaphase, and telophase, it starts to split. And then after cytokinesis, we end up with 2 daughter cells. Not only does it produce 2 daughter cells, but those daughter cells are identical. They're identical to each other, and they're also identical to the parent cell. So this is a good image that shows that. Our parent cell starts off with this red DNA and this baby blue DNA. And after our process of cell division, we end up with 2 daughter cells that have that same red DNA and that same baby blue DNA. And they have the same DNA as each other, but also the same DNA as the parent cell. So mitosis produces identical daughter cells. Additionally, those daughter cells are diploids, which means they have 2 complete sets of chromosomes. Diploid cells. Of course, we've got the prefix di, the prefix di in that word diploid. So that represents the word 2. Di means 2. We've got 2 complete sets of chromosomes, 1 from each parent. Mitosis is used to produce somatic cells, body cells. So those would be your nerve cells, your muscle cells, your digestive cells, or your skeletal cells. Basically all of your cells except for your sex cells. And lastly, mitosis is used for asexual reproduction. Solo dolo. You only need 1 organism or cell for asexual reproduction. Okay, any questions about that process? Thank you, Angel.
No.
Thank you, Ayana. So when we get to the end of this presentation, we're going to compare mitosis and meiosis. Maybe some questions will come up then, but hopefully you all are pretty clear on these processes so far. Okay, our second and last standard for Unit 3 is Standard 3.2.1. It's about meiosis. Explain the role of meiosis in sexual reproduction and genetic variation. So, characteristics of meiosis. Meiosis has 2 rounds of prophase, metaphase, and anaphase. There's meiosis 1 and meiosis 2. Prophase 1, metaphase 1, anaphase 1, telophase 1. Then again, prophase 2, metaphase 2, anaphase 2, and telophase 2. So we've got 2 rounds of PMAT. Meiosis produces 4 daughter cells. And each of these daughter cells is unique. So again, we've got that same general setup with our baby blue DNA and our red DNA. But you can see that in addition to producing 4 cells instead of 2, each of them is a little bit different. We've got some red DNA on this blue chromosome and then another blue chromosome. We've got some red DNA on this blue chromosome with a red chromosome, so on and so forth. There are slight differences between the two. Or I'm sorry, between all 4 of the daughter cells. There are differences. So each one is unique. And each one is also called a haploid cell. Haploid means that it has half of a complete set of chromosomes. So our body cells, our somatic cells, have 46 chromosomes, but our sex cells have 23 chromosomes. And this ensures that when 2 sex cells meet to begin the process of creating a human being, a baby, they add up to the total 46 chromosomes, if everything goes well. Meiosis is used to produce gamete or sex cells. So these would be our sperm and egg cells. And lastly, we can think of meiosis as being sexual reproduction. It takes 2 to tango. You need 2 cells for sexual reproduction, a sperm and an egg. Okay, any questions about meiosis?
Wow.
Thank you, Ayana. Thank you, Kiki. Okay. Again, if anything comes up, you can pause me or stop me at any time. So here's looking a little bit more into meiosis and exactly how we get into these 4, how we result in 4 genetically unique daughter cells. In any case, we're starting off with a parent cell that is a diploid, and it's a somatic cell. So it's 2N. That's what diploid means. It's got the full 2 sets of chromosomes. So the DNA has now been replicated and it's condensed into chromosomes. The next step is that they would cross over. So you guys may not have caught that, but look at the chromosomes here. We've got one chromosome that is entirely red, one that is entirely blue, one that's entirely purple, and one that is entirely orange. But watch this. They do what's called crossing over, which means that homologous chromosomes, meaning chromosomes that are the same size, exchange just a little bit of their DNA. So these 2 chromosomes are the same size, the red and the blue ones, so they exchange a little bit of their DNA. These 2, the purple and the orange one, they also do what's called crossing over, and they exchange some of their DNA. So quickly you can see it. They're just crossing over here. They're just exchanging some DNA. And this is an important process because it contributes to diversity. And you're going to see exactly how that happens in the next couple slides. But you can literally see how it's just crossing over, crossing over. This happens during prophase 1. Not super important that you know that, but I just wanted to represent and display what crossing over looks like. It's just an exchange of DNA between homologous chromosomes, chromosomes that are the same size and contain the same genes. All right, so after the first round of meiosis, after meiosis 1, we've gone through prophase 1, metaphase 1, anaphase 1, telophase 1. Now we end up with 2 daughter cells. They're still both diploids, but they're already genetically unique. So we can see in this original parent cell, we've got red, blue, purple, orange. Over here we've got red and orange with a little bit of purple and a little bit of blue. And over here we've got blue and purple with a little bit of red and a little bit of orange. So already we've got 2 daughter cells that are already genetically unique. They're not the same anymore. And then lastly, after meiosis 2, so after the second round of prophase, metaphase, anaphase, and telophase, we end up with 4 daughter cells. And not only is each of them a haploid, but they're also still genetically unique. So over here, I've got an entire chromatid that is red. But over here, I've got a chromatid that's partially red and partially blue. And over here, I've got a chromatid that's partially blue and partially red, and so on and so forth. So each one of these is a little bit different than the other. And that's all the results of this original crossing over. That crossing over process is extremely important, and it contributes to this genetic diversity. It means that each of our sex cells is a little bit different than the other. Now, I simplified it to only use 4 colors: red, blue, purple, and orange. But in reality, it's not colors that matter, it's DNA. And there are many thousands, millions of different combinations, billions of different combinations that can produce totally unique sex cells. We can also notice that they, instead of having full chromosomes here, they only have chromatids. So they're now haploids. They have half the amount of DNA. Okay, so that explains some of the genetic diversity, but there are other sources of genetic variation. And as we got into a pretty good conversation on Friday about genetic variation and its importance. And we talked about it's important because it increases chances of individual survival, but it also increases chances of species survival because you can pass on your genes to the next generation. So it's not just about individual survival, it's also about species survival. And genetic variation is a great way to protect species survival. We talked about 5 sources of genetic variation, and I'm not going to cover them in great detail right now. But those 5 sources were random assortment, crossing over, which we just discussed, mutations, non-disjunction, and fertilization. Random assortment and crossing over are guaranteed to happen always. They always happen. Random assortment means that it's totally random. You can't predict which chromosome will end up in which sex cell. There's just no way to predict that. And crossing over is that process that we just looked at where homologous chromosomes exchange some of their DNA. So those 2 happen all the time. Mutations, non-disjunction, and fertilization are not guaranteed. They are pretty common, but they're not guaranteed. Okay. All right, so I believe this is it. This is, after this, we will maybe play a Kahoot, but let's see, can we classify all of these different terms as being characteristics of mitosis or meiosis? So the first term we have here is sexual reproduction. Is that mitosis or meiosis?
Meiosis.
That is meiosis, good. Excuse me, what about diploid cells? Would those be mitosis or meiosis?
Mitosis.
Good, that's mitosis. 1 round of PMAT. One round of PMAT. What do we think?
Mitosis.
That's mitosis as well. Gametes,
Meiosis.
good. 2 rounds of PMAT,
Meiosis.
good. Sex cells,
Meiosis.
good. Asexual reproduction,
Mitosis.
excellent. Haploid cells,
Meiosis.
good. Body cells,
Mitosis.
excellent. Sperm and egg,
Meiosis.
4 daughter cells,
Meiosis.
good. 2N,
The one that starts with an MIT.
good, good. So 2N is another way of saying diploid cells, just to clear that up. 2N is another way of saying diploid. 2 daughter cells,
Mitosis.
excellent. And then 1N,
Meiosis.
good. So these are some clear differences between the two processes. You definitely want to be aware of these. And if you can keep these in mind, these differences, then a lot of the questions on tomorrow's exam will be pretty easy to you. Also, this is a good opportunity for me to mention that our exams, as we know, are cumulative. So please make sure you refresh your Unit 1 and Unit 2 notes. Just, I'm not saying 2 hours on each unit or anything like that, but just quickly look through them again and make sure that you remember some of the foundational concepts. Of course, both of these processes can only occur after DNA replication. That's so important to keep in mind, guys, because some of the questions might try to trip you up and say, what could happen? Which one of these is more likely to happen if the DNA is never replicated? Mitosis, meiosis, or neither, right? So that's not an exact question from the exam, but you must know that DNA replication has to occur before either one of these M phase processes. Okay. So at this point, we'll play this quick quiz. I said Kahoots. Let's do this. Let's do this one. Oh, it is a Kahoot. And it's 17 questions. Okay, hold on. That was bad. All right, so Kahoot.it. Sorry, Angela, I didn't see you responding in the chat. Kahoot.it, and then the code is 738-7506. Thank you, Kiki. Good, we've got 7, 8 folks. We've got 12 on the call. Let's see. Okay, I'm going to wait 30 more seconds. I think we've got 4 people who have not yet joined. All right, let's go ahead and get started. Name this phase. Good. All right. So again, the giveaway is that the chromosomes are lined up in the middle. Meta means middle. Metaphase represents chromosomes lining up in the middle. Good job. Yeah, that was somewhat of a tough one. There was no media, right? So, yeah, cytokinesis again is when the cells split into 2 new cells. In order for a cell to be considered, its own cell, it needs to have its own cytoplasm and its own plasma membrane. That was a tough one, though. Good job, Alicia. Good. Good job. We've got some big jumps. After mitosis, how many cells are there? Good. So mitosis produces 2 daughter cells. 2 daughter cells. Good job. Again, tough one. Not stuff that we've actually explicitly covered, but good job, Amari and Alicia. Good, anaphase. So we can see that the chromosomes have been pulled apart. Ana means apart, so anaphase it is. Yep, we can see this dotted line. And as Ayana told us, and Alicia as well, when that dotted line is there around the nucleus, that means that the nucleus is either dissolving or it's reappearing. But in this case, we know that it's dissolving because we see chromosomes here. And this is clearly before the nucleus has divided. Good. So, okay. So meiosis, of course, as we go through 2 rounds of PMAT. So PMAT, PMAT, that's 8 phases. Good job, Amari. Good. Just talked about that one. Good. Excellent. Sex cells are haploid, so they have half. Good job. What comes after metaphase? Anaphase. Good job. Okay. Good. DNA gets replicated in synthesis. Very, very important stuff right there. Good. Was review of Unit 2. Which organic compound is DNA? This is Unit 1. Excellent. The Na in DNA stands for nucleic acid. Good job. Glad no one got that one wrong. During which phase of cell division do the chromosomes and spindle fibers begin to appear? Prophase. Excellent. And last question. Which phase of the cell cycle is considered the longest and why? Beautiful. Interphase. Let's see. Excellent. Okay. Good job to those three. Good job to everyone. Let's see. New game. I have that set up for my next class. All right. So at this point, there is an exit ticket, but beyond that, there is no assignment. Your assignment is to study for our exam tomorrow. And I'm going to make sure that I do my best to work out the technical kinks. Okay, so any questions? Now is the time to ask. So we have 11 minutes left and we can go over anything you'd like to go over.
I have a question about the test. Is it going to be on SchoolNet again?
Not likely. I'm probably either going to use Canvas or Mastery Connect, depending on what the other teachers want to do.
That sounds like that will work better than last time.
Agreed. Neither one of them works, huh, Jalen? Okay, let's see. Did anybody else have that same issue? What the? My laptop hasn't been plugged in this whole
Mr. Rudd,
time. Yes,
is the exit ticket is just that thing, right? That's it. Like it just asks you how I feel about the test.
yes.
Okay. All right, Mr. Rudd.
Okay.
Mr. Red,
Yes.
is there any since you said there's no assignment, just at the exit ticket, is there any big assignments that I missed while I was out?
Well, everything is on Canvas. So if you check Canvas, you should be able to see what you've done and what you haven't at this point.
Oh, okay. Yeah, this past. Oh yeah, and sorry that I didn't really show up this past week. I was really sick, so yeah.
Okay.
There's nothing like any test that I need to like put priority first.
No, I don't think so.
Okay. That was it.
Let's see. Joseph, when are you updating grades? I likely will have it done by the end of tomorrow when we finish with the exam. Let's see this. I'm also going to upload the notes to Canvas.
But that's all for today, right, Miss Rudd?
Yes.
Oh, all right then. I'll see you tomorrow then.
Okay, see you tomorrow. Yes, Olivia, you're good to go.
Bye.
See ya.
We can leave.
See ya. Have a good day. Yes, you're good to go.
Okay. See you tomorrow.
Thank you. See ya.